Hey guys, welcome to the new video. Today I'm gonna cover all the chapters for exam 2 for human physiology. And so yeah, let's just go over it. Now, chapter 4 covers neurophysiology, so yeah, let's just go over it. Okay. The resting membrane potential. The resting membrane potential is also known as the polarized state. So in a neuron, the standard resting potential is approximately negative 70 millivolts, but it can range from negative 40 to negative 90 millivolts. So basically what it's trying to say is the polarized state is negative 70 millivolts in a neuron. And this is when the neuron is not actively sending a signal. It is positively charged outside and negative inside, meaning there's high sodium outside and high potassium inside. So if you look at the drawing here, if you look at the inside of a neuron, there's high amounts of potassium, but there's low amounts of potassium outside. But if you look at the outside right here, there's high amounts of sodium, but low amounts of sodium here. And this is going to be important in a bit because this is what causes depolarization and all those processes that goes afterwards. Measuring potential differences between 2 points in a neuron. So this image here basically just discusses the potential difference between 2 points in a neuron. And as you can see here, there's one microelectrode outside and one microelectrode inside. So what it's saying here is that the charge in the inside is negatively charged relative to the outside, which we know from here because it literally says it's negatively charged inside. Okay, nerve and muscle tissues are excitable. Excitable meaning that it responds to electricity, and they can undergo rapid changes in their membrane potential. So they can change their resting potential into electrical signals. So in this case, nerve impulses for nerves and muscle contraction for muscles. And these tissues can rapidly change their membrane potential, transforming resting potentials into electrical signal. Okay, nerve and muscle cells can develop several electrical states at their membrane. So we first have polarization. Again, polarization, this is any state, positive or negative, other than 0 millivolts. In this case, for a neuron, it's negative 70 millivolts. That's all you have to know for polarization. This is when the cell is not actively sending a signal. Then let's say a stimulus happens. This causes a depolarization effect on the neuron. Now, depolarization, this makes the membrane potential less negative than the resting potential. So what happens is, in depolarization, sodium goes in and becomes more positive. So again, if you guys remember from the image that I drew, there's high amounts of potassium outside and high amounts of sodium outside. So what happens is in depolarization, sodium ions go into the neuron, and this makes the inside of the neuron more positive. And this is why it goes up like this, because sodium is making the neuron become more positive. Then we have repolarization. The membrane returns to the resting potential after depolarization. So after a certain amount of depolarizing, which is typically around 30 millivolts in this case, repolarization occurs, and this is when potassium goes out and it returns it back to negative resting membrane potential. So again, right, sodium goes in, right? This makes the neuron more positive, but now potassium is leaving the neuron, and this makes it more negative, and it returns it back to the resting membrane potential. Then we have hyperpolarization, and this makes the membrane more polarized, meaning it makes it more negative inside. So after repolarization around this area right here, as you can see, it goes down below to the negative 90-ish millivolts, and that's because it hyperpolarizes, and this is usually the cause of more potassium leaving, as you can see here. So after potassium leaves, right, and it goes back to negative 70 millivolts, usually potassium ions are bigger, so these channels right here, they take a little bit longer to close. So what happens is extra potassium leave, and that can cause hyperpolarization, or chloride ions accidentally go in, and this makes it more hyperpolarized. So there's two causes into hyperpolarization, and just know that chloride or potassium makes it more negative. Right here, see? Okay. Electrical changes can occur by the changes in ion permeability and movement across the plasma membrane of nerve and muscle cells. So this is basically what I just said in here, but in wording. So as you can see, a flow of positive ions into the ICF depolarizes the cell. The positive ion is referring to the sodium going in, right here, see? Sodium going in. A flow of positive ions into the ECF can repolarize a cell after depolarization. So this is referring to the potassium leaving the cell. As you can see here, it's leaving the cell, and this repolarizes the cell after depolarization. And from resting state, a flow of positive ions into the ECF can hyperpolarize the cell. This is basically saying extra potassium are leaving. Because again, if you guys don't know, potassium ions are bigger than sodium, so these channels are a bit bigger, so they take a bit longer to close compared to sodium channels. So extra potassium leave even after potassium channels reaches negative 70 millivolts. Type of signals that can be generated. We first have graded potentials. This can be altered. Okay, so graded meaning different levels of power. So there can be little, a lot, or it can be brief or long. So just know that graded potentials can be altered, and it is mainly for short distance. For action potentials, they can't be altered. They're all identical, and they are made for long distance. So in this case, for example, guy stubs his toe, electrical signals to the axon hillock. Axon hillock triggers action potential right here. And the axon hillock goes from the toe to the lower back to spinal cord to the brain. So just know that action potential is for long distance and graded potential is for short distance. And that graded potential can be altered, whereas action potential can't be altered. They're all identical. Okay, so changes relative to the resting membrane potential, depolarize. Again, this is what I just mentioned over here. It's saying that reduced potential, sodium goes in, increased potential, more potassium out or chloride in. Depolarization and hyperpolarization of the membrane. This is basically what I just said again. As you can see, that rise in that stub right there, that's the sodium going in. That's depolarization. And then after it reaches resting potential, it hyperpolarizes, sorry. And this is usually the cause of potassium leaving or chloride ions going in. A graded potential is local, meaning it is short distance. Electrical change in the membrane. Now, it occurs in varying grades. This change is progressively greater as the triggering event becomes progressively stronger, and it is usually the cause of a flow of sodium ions into the ICF that produces this kind of potential. And its duration of graded potential is directly proportional to the duration of the triggering event. So in other words, slide 10 and 9 is basically saying that it's emphasizing the nature of graded potentials as localized changes in the membrane potential that spreads passively and decrease in strength over distance. Because again, they're short distance, right? This makes them fundamentally different from action potential, which is actively propagate without diminishing in strength. And the spread and influence of graded potentials are crucial in determining whether a neuron will fire an action potential, thereby affecting how signals are processed and transmitted in the nervous system. So yeah. All right, graded potentials die out over short distances. Again, we know that they're short distance. Their spread is decremental. And for examples, graded potentials are postsynaptic, receptor, end plate, pacemaker, and slow wave. So just know that any word that ends with potential, in the exception of action potential, are graded potential. So for example, postsynaptic potential is a graded potential. Receptor potential is a graded potential. End plate potential is a graded potential. Pacemaker potential are graded potential. But in the exception of action potential, it is not a graded potential. Graded potentials converge on the axon hillock to generate an action potential. So what we have here is a stimulus. Right here is the stimulus. This creates a graded potential. Again, graded potential, they're short distances and they can vary in grades. Grades referring to the different levels of power. So it can be strong or it can be weak, it can be long or it can be short. So as you can see here, the graded potential travels this way to the axon hillock. Now at the axon hillock, it will generate action potential if it has enough energy and reaches the threshold. The threshold that the amount of energy needs to generate action potential is negative 55 millivolts. So let's say the graded potential has enough energy. What happens is it goes like this, right? Graded potential has enough energy. It reaches negative 55 millivolts first, and then it will generate an action potential. Now this causes a depolarization in the cell. But then let's say that it doesn't have enough energy. The graded potential doesn't have enough energy. This causes a subthreshold potential. So what we have here is a stimulus. It goes up, but it doesn't reach negative 55 millivolts, so it will fall down. And this is called the subthreshold potential because it does not have enough energy to generate action potential. Threshold and all or none phenomenon. So this is referring to the action potentials either occurring or they don't. And there's no partial action potential. So as long as the graded potential reaches negative 55 millivolts, then it will generate an action potential. Graded potential may be subthreshold. So again, this is referring to it not having enough energy to trigger an action potential. And the stimuli varies in the amount of current and the number of signals received in a given amount of time. So an action potential occurs if a membrane reaches the threshold potential, which is again negative 55 millivolts. The voltage gated channels in the membrane for sodium and potassium ions undergo conformational change, and the flow of sodium ions into the ICF reverses the membrane potential from negative 70 to plus 30 millivolts, and the total charge is about 100 millivolts across the membrane during an action potential. So this is referring, these dots right here, they're referring to depolarization. Again, we have a stimulus here, we have a stimulus right here, right? It doesn't reach negative 55 millivolts. This is called subthreshold. So then let's say again, we stimulate it again, but this time it reaches negative 55 millivolts. This will cause depolarization. And depolarization is when it reaches the threshold. It stimulates both sodium and potassium channels, but sodium channels open faster than potassium channels. So basically think of sodium channels as a motorcycle and potassium channels as a truck at a stoplight. So at a stoplight, you would expect sodium ions, because they're a motorcycle, right, to go faster than the truck. So even though they're given the green signal at the same time, they don't move at the same pace because motorcycles, they are lighter in weight compared to trucks. So depolarization, this is when the sodium flows inside the cell. As you can see here, sodium goes in and all that. Then after a short amount of time, it depolarizes above all the way to plus 30 millivolts. And after plus 30 millivolts, the sodium channel inactivation occurs, and this is when sodium gate finally closes. And this is when potassium ion channels is actually opened. So again, sodium channels and potassium channels, although they were given the same green signal to go and open at the same time, potassium channels, they open slower compared to sodium channels. And that's because potassium channels are bigger than sodium channels. And this is referring to the 100 millivolt change. So again, the resting memory potential is negative 70 millivolts, right? It reaches plus 30 millivolts right here. That is a 100 millivolt change. Then we have repolarization. This is when the sodium channel is finally closed, and this is when the potassium ion channels are finally open. And this causes a flow of potassium outside the cell. So as you can see here, they leave the cell. And this brings it back to negative 70 millivolts. However, now that we go back to negative 70 millivolts right here, the sodium potassium ion channel inactivation gate resets. But during that process, potassium channels are still closing because again, they're bigger compared to sodium channels, right? So they take longer to close. And this causes hyperpolarization. This is when more potassium leaves, or chloride accidentally gets into the cell. This hyperpolarizes the cell. So now at negative 90 millivolts, this is when the potassium gate also closes. And then in this process right here, this is when the resets occur. Bringing it back to negative 70 millivolts. I'll explain more in a bit if that doesn't make sense. Generation of action potentials. There's 3 overlapping steps. We first have the transient increase in sodium permeability. Again, this is depolarization right here. It depolarizes the neuron. Then we have sodium impermeability. This is referring to the sodium gates finally closing and potassium gates finally opening. And this causes repolarization right here. And so yeah, this is basically what I just said in the couple minutes I've been talking. Oh, again, this is repeating itself. Depolarization, increased sodium permeability, and reverse membrane potential. But the gate must reach a threshold, then become self-generating. So this is a regular neuron. This is polarization when the neuron is not actively sending a signal. So then let's say a graded potential comes in. This causes, a graded potential comes in and it has enough energy to generate an action potential. This causes the sodium channels to finally open. And the sodium goes in until it reaches a plus 30 millivolt charge. Okay. Now at 30 millivolts, the sodium gates finally close and potassium gates are finally opened. And potassium gates are opened until it reaches about negative 90 millivolts. So I guess this kind of skipped the repolarization and it goes to hyperpolarization. Now between negative 70 millivolts to negative 90, so this is referring to over here, the undershoot, sodium channels sneak out, and then sodium and potassium pump resets to negative 70 millivolts. So now we're back at negative 70 millivolts. Now, resetting the system. This is referring to this stage right here. This is negative 90, right? Now to get back to negative 70 millivolts in this area right here, we need a resetting system. And this is called the sodium/potassium pump. They are revved up following the repolarization stage, which resets the original concentration of ions. Only a few ions need cross to change the permeability. Again, I already went over this. The rising phase is referred to as depolarization, and this is when sodium influx happens. Then at plus 30 millivolts right here, we then have repolarization. And this is usually the cause of the efflux of potassium. Again, this is basically saying potassium leaves. And then right here we have the undershoot. The undershoot is the extra potassium leaving or chloride ions going in. And then around this range right here, we have the reset, the pump reset. This is when this is when it resets the original concentration of ions it needs to get back into polarization. This is basically what I just said in this image. They're the same. All or none and the coding for the stimulus intensity. So either an action potential occur or the threshold is not reached and no action potential is generated. This is basically saying the all or none. Action potentials either happen or they don't. Once generated, the intensity does not vary because all action potentials are identical. It is the rate of action potentials that are given that determine the intensity of response, and the frequency of the impulse determines the strength. So this is referring to the action potentials. Action potentials, again, they're long distance and they're all identical. They can't be altered. So if they can't be altered, you can, however, change the frequency of how action potentials happen. So for example, within a span of 0 to 10 minutes, if only one action potential happens, then you can say that the signal is not that strong. But then let's say within a span of 0 to 10 minutes and 7 action potentials occur, then you can say that it is somewhat strong. Then now if we have 26 action potentials within the span of 10 minutes, then you can actually say that it is really strong. So the frequency of the impulse determines the strength of the action potential. Okay, so again, right here, what we have here is negative 70 millivolts. If it doesn't reach negative 55 millivolts, this is called the subthreshold potential. And it doesn't reach, that means it doesn't have enough energy to cause an action potential. So then, now let's say we have a stimulus right here, it reaches negative 55 millivolts, and look at the action potentials. In here, there's 7 action potentials, and there's an increase in amplitude. Then right here, we have another stimulus. Now we have a bigger amplitude because there's 9 action potentials. And then again right here, stimulus right here. Now we have an even bigger amplitude, and that's because there's 11 action potentials. Now, action potentials travel from the axon hillock to axon terminals. So again, let's review on what we just went over. Again, we have a stimulus right here. This causes a graded potential, and the graded potential goes to the axon hillock. Now at the axon hillock, if the graded potential has enough energy to reach negative 55 millivolts, this will cause an action potential right here. And the action potential will travel along the axon to the axon terminals right here. So that's basically what it's saying right here. Once initiated, an action potential is conducted throughout the axon. A refractory period ensures a one-way travel of an action potential, and it also limits the frequency of an action potential. So we first have the absolute refractory period. This is the period right here in the middle. It's kind of light. And this is when you can't fire another action potential on top of another action potential. However, you can increase the frequency of an action potential, so they can increase the amplitude of it, basically. Then we have the relative refractory period. It is possible to fire another action potential, but it requires a stronger than normal stimulus. So the relative refractory period refers to this stage right here. However, you need a stronger stimulus because at this period you're at negative 90 millivolts, right? Now you need a plus 25 millivolt of a stimulus to trigger an action potential. Now in a regular action potential, you only really need to reach plus 15, right, to reach negative 55. Well, now you're at 90 millivolts. So now you need a 25 millivolt change or a stronger stimulus to trigger another action potential. And you can only do this during the relative refractory period. Okay. So conduction velocities in axons, and the speed varies widely among neurons, and they can move up to 100 feet per second or 30 meters per second in meters, right? 100 feet per second or 30 meters per second. They're the same thing. And these depend on 2 factors. We first have the axon diameter. So in axon diameter, the larger the diameter, the faster the conduction. And we have the degree of myelination. The degree of myelination refers to these little sheath right here. So if it was unmyelinated or there's no myelination, it is a continuous conduction, and it is relatively slow because the entire axonal lemma must be depolarized. So this refers to this graph right here. So let's say a stimulus happens right here. So what happens is the entire axonal lemma must be depolarized. So it'll jump from here to here to here to here to here for about 30 times. And the number 30 right here, this is there's no significance. It's merely for comparison. So I'm just saying that in unmyelinated axons, the entire axonal lemma must be depolarized. Now, if it was myelinated, what happens is a stimulus happens right here. It would perform a thing called the saltatory conduction, and it'll jump from nodes of Ranvier to the other. So, for example, it'll be right here, right? Stimulus happens, it'll go from here, and then it'll jump from nodes of Ranvier to nodes of Ranvier to nodes of Ranvier right here, and it'll jump about 5 times, or about 7.5 times faster than unmyelinated. The nodes of Ranvier, by the way, are these little spots right here where the axons go down to jump into. So unmyelinated axons, they're merely for processing, and you can find them in gray matter. For myelinated axons, they're mainly for speed, and they're found in white matter. So I'm pretty sure you guys already took anatomy. I'm pretty sure you guys get the gist of gray versus white matter. We'll get more into that information once you get into the central nervous system part of the video. But right now, all you really need to know is that unmyelinated axons are for processing, which therefore are relatively slower compared to myelinated axons, in which they are made for speed, and they can be found in white matter. So that's all you really have to know in here. Okay, so we have saltatory conduction. Again, this refers to the action potentials jumping from nodes of Ranvier to the next. So again, at the axon hillock, if a graded potential has enough energy, it triggers an action potential. And the action potential jumps from nodes of Ranvier to nodes of Ranvier to nodes of Ranvier, and that is called the saltatory conduction or the jumping conduction. In the peripheral nervous system, you can find it in in the peripheral nervous system, they are named Schwann cells. So this example right here that I went over, these right here were Schwann cells. And in the central nervous system, they are known as the oligodendrocytes. And they can wrap into multiple axons, but in this picture, it only showed two. But yeah, more information into that once you get to the central nervous system of the video. Myelination with nodes increases the speed of conduction of the action potential. So again, this is basically what I just said in this part. The action potentials is generated from the axon hillock right here. It will jump from nodes around the ear to nodes around the ear to nodes around the ear and nodes around the ear. Just know that it is myelinated and it is faster than it being unmyelinated. The oligodendrocytes form the myelin in the central nervous system, and the Schwann cells forms the myelin in the peripheral nervous system. So let's go over what we just went over right now from literally slide 1 to now. So what we have here is a neuron. The neuron has a bunch of dendrites. It has an axon hillock and it has the axon. Then it converges into axon terminals, like at the end right here. So what we have here is an axon. Now, if there's a stimulus, let's say a stimulus right here, this creates a graded potential. The graded potential then goes to the axon hillock. At the axon hillock, if it has enough energy to generate an action potential, which is around negative 55 millivolts, if it has to reach that point, it will generate an action potential right here. Let's make this neuron myelinated right here. So these right here are Schwann cells. So let's say the action potential is generated from the axon hillock. Then it will jump from nodes around the ear. The action potential will jump from nodes around the ear to nodes around the ear to nodes around the ear to the next until you reach the endpoint of the axon terminals right here. Of course, it'll be like that too if it was unmyelinated, but it'll take a bit longer if it was not myelinated. So that's what we went over the couple slides that we were just talking about, right? So nerve fibers can regenerate. So Schwann cells in the peripheral nervous system guides the regeneration of cut axons, and the regeneration of cut axons in the central nervous system is inhibited by oligodendrocytes. So in other words, the summary of the slide is saying, or it's highlighting the significant differences in nerve regeneration capabilities between peripheral and central nervous system. While the peripheral nervous system has mechanisms and cellular support for effective nerve regeneration, the central nervous system presents substantial challenges that currently limit the recoveries from injury involving spinal cord and brain neurons. And this information is crucial for understanding the complexities of nerve repair and the development of treatments aimed at enhancing nerve regeneration in the central nervous system. Okay, so what happens when two neurons come together? Well, you get a synapse or a gap. There's two types. There's electrical and chemical. So let me give you guys a good analogy explaining this image. So imagine you are cold. Now in your skin, you have a bunch of receptors that basically detects the temperature of your surrounding. So right here's your skin and there's a bunch of receptors. Now let's say that the receptors in your skin determine that you are in a very cold environment. So the information that is being sent to the brain is the afferent division because it is sensory. It senses something and sends information into the central nervous system. Okay, so then let's say because you are cold, you decided to wear a sweater. Now, before all of that happens, your central nervous system or your brain has to send signal through the peripheral nervous system to the efferent division. The efferent division is also known as the motor division because it controls an effector organ, and that can be a muscle or a gland. So because you wore a sweater, the efferent division or the motor division sent signals to the effector organ to move and wear a sweater. So that's basically your muscles moving to wear a sweater, right? Now, in the process of sending information from a neuron to a neuron, we have a gap or a synapse right here. There's also one here too. By the way, this image right here, we're going to go more information into that later in the video. Okay, so right now, all we have to know, or all we have to really learn right now is the gap or the synapse of when a neuron comes together. So there's two gaps, right? We have a chemical and a electrical. Electricals, they are rare because cells are electrically coupled as cytoplasms of adjacent neurons are connected, and this allows synchronization of neurons. So what happens is action potential comes here, and they can just go straight through like that. And this is basically for speed, but there's no control. Then we have chemical. Now, chemical is the one that we have to focus on because it is the most common. This image right here, this is just a simple overview, but I'm going to explain more in detail right here. So this and this are basically the same thing, except this right here is more detailed. Okay, so I want you guys to focus on this thingy right here, this pointer thingy, while I'm reading the explanation as to what's going on. So we first have presynaptic. What happens is, in the presynaptic, again, we have an action potential. The action potential opens the calcium voltage-gated channels, letting calcium into the axon terminal. Once the calcium goes in, they bind to the proteins or the neurotransmitter. The proteins take the vesicles that stores neurotransmitter and cause them to move by exocytosis into the plasma membrane of the axon terminal. Acetylcholine is the neurotransmitter that was binded by the calcium. So once the acetylcholine is in the plasma membrane of the axon terminal, they bind to the receptors of the ligand. Okay. Then these channels that were binded by the acetylcholines are sodium channels. So sodium goes in, depolarizing the cell, causing an EPSP right here. And an EPSP is a graded potential. Again, let me remind you, EPSP, they are for short distance. So again, EPSP is generated from the sodium going in from the ligand channels right here. The EPSP spreads outwards to the end plate, opening the sodium voltage-gated channels on the other side of the neuron. So right here, these channels right here, these are voltage-gated channels for sodium. So once the EPSP opens these channels right here, sodium goes in, depolarizing the whole neuron again, triggering another action potential. And now you would need acetylcholine esterase or HAs to break down acetylcholine to deactivate the communication of one neuron to another neuron. So yeah, that's basically the whole point of the couple slides that I'm going to go over. So again, slide 33 says a neurotransmitter can carry a signal across the synapse by a series of steps. An action potential at the axon terminal of presynaptic neurons open calcium channels. This refers to this step right here. And then calcium flows into the ECF through the channels into the synaptic knob right here. The calcium influx induces the rupture of presynaptic vesicles in the axon of the presynaptic neuron right here. And then we get a neurotransmitter that can carry a signal across the synapse by a series of steps. These vesicles release a neurotransmitter molecules which enter the synaptic cleft by exocytosis. So these neurotransmitter molecules is referring to the acetylcholine right here. Okay. These molecules diffuse and bind to the receptors of the subsynaptic membrane of the postsynaptic neuron. So these right here are subsynaptic membrane or the ligand channels. Okay. These binding open the chemically gated channels in the subsynaptic membrane, leading to the events that excite the postsynaptic neuron. So this is referring to the sodium going in and causing a small graded potential of EPSP, which triggers another voltage gated channel, sodium channel, to go in, causing an actual action potential. Okay. This is basically what I just said in here. So IPSP and EPSP. EPSP allows sodium and potassium to diffuse simultaneously, eliminating the threshold. This generates numerous graded potential to reach the axon hillock. We then have IPSP. IPSP hyperpolarize the membrane by increasing the permeability of potassium or chloride. This makes reaching the threshold, hence the action potentials, much more difficult. So the other characteristics of neurotransmitters. They are quickly removed from the synaptic cleft. Some function through the intracellular second messenger systems. Presynaptic inputs determine a postsynaptic potential by temporal summation and spatial summation. By temporal summation, EPSP occur close together in time from a single presynaptic neuron, and by spatial summation, EPSP originate from several presynaptic inputs. IPSP can also undergo temporal and spatial summation. So in other words, slide 38 basically provides an insight into the dynamic and complex regulation of neurotransmitter activity at synapses, highlighting the mechanisms to ensure precise communication between neurons. The rapid removal of neurotransmitters and their modulation through second messenger systems are crucial for maintaining the efficacy and precision of synaptic transmission. Understanding these processes is key to comprehending how the brain adjusts its responses to various stimuli and maintain homeostasis. So, neuronal integration of EPSP and IPSP at the axonal membrane of the postsynaptic cell. So we first have subthreshold. There is no summation. Again, subthreshold, this is when the graded potential does not have enough energy to reach negative 55 millivolts to trigger an action potential. So as you can see, it doesn't reach negative 55 millivolts, so there is no summation. We then have temporal summation. Temporal summation is when it fires twice in a rapid succession. So as you can see here, it triggers a graded potential that is not enough to reach negative 55. However, it's going to fire again before it goes down. And then now it reaches negative 55 millivolts and therefore creating an action potential. So that is temporal summation. It fires twice in a rapid succession. And then we have spatial summation. Spatial summation is when you have two signals from two different areas firing at the same time. So right here, what happens is two signals from two different areas. They trigger an EPSP, one right here, and it reaches negative 55 millivolts, therefore causing an action potential. And then we have spatial summation of EPSP and IPSP. So the IPSP again is inhibitory, whereas EPSP is excitatory. So what it's saying is that they cancel each other out. So right here, it goes down because it's inhibitory, right? But right here, there's excitatory. So basically nothing happens because they cancel each other out. And yeah. So neurotransmitters. Neurotransmitters are the chemicals used by the nervous system to send messages to the rest of the body. There's over 50 types that have been identified, and they are classified in two ways: chemically or functionally. So, chemical classification of neurotransmitters. We first have acetylcholine, and these can be found at the neuromuscular junction. This is the most common, and they control muscles. We then have biogenic amines. These include epinephrine, serotonin, broadly distributed in the brain, used in emotion. So they're basically used for regulating mood, arousal, and sleep. We then have amino acids, GABA or the gamma aminobutyric acid, and glycine in the central nervous system. We then also have peptides. Peptides are endorphins for pain and digestion. And then we have novel messengers, the ATP. So we have nitrous oxide and carbon monoxide, systemic and local transmitters. Incredibly complicated active area of research. Neuropeptides act mainly as neuromodulators. So they are molecules consisting of 2 to 4 amino acids, and they do not produce EPSP or IPSP. They modulate at the synapse, bringing about long-term changes. So the effectiveness of the presynaptic input can be altered by the presynaptic facilitation or inhibition. The presynaptic facilitation, this is when there is enhanced release of neurotransmitter from the presynaptic axon terminal, whereas the presynaptic inhibition is the reduce of releasing the neurotransmitter from the presynaptic axon terminal, and they are each influenced by another nearby axon. So for facilitation, this increases the release. So for example, to increase the release of neurotransmitter, you can increase the frequency of action potentials. You can increase the calcium going into the axon terminal. You can increase the enzyme. You can increase the exocytosis. And then for inhibition, you can decrease the release by decreasing action potential. You can decrease the calcium going in. You can decrease the ATP. You can decrease the number of receptors over here. You can increase acetylcholine esterase, which basically breaks down the acetylcholine that's typically released by the neurotransmitter and all that. So yeah, just know that presynaptic facilitation enhances the release of neurotransmitter, whereas presynaptic inhibition reduces the release of neurotransmitter. Okay, so drugs and diseases can alter the synaptic transmission. So for example, cocaine, they block the reuptake of dopamine. Parkinson's disease, this is due to the deficiency of dopamine in the brain region controlling the complex movements. We have strychnine, which competes with glycine, which is an inhibitory neurotransmitter, and tetanus, which the toxin prevent the release of GABA, which affects the skeletal muscles. Neurons are linked through a complex of converging and diverging pathways. So by converging input, a single cell is influenced by thousands of presynaptic cells right here. And by divergence, a branching axon terminal affects thousands of postsynaptic cells. And that should be it with the video.